make sure it works. Okay, I think we're getting started. Thank you guys. Thank you to the Alliance for inviting me to give a talk again this year. I have a lot more data than we've had previously as we've been working with this new model, the new model that Scott Doe and Larry Reiter created, and a lot of the data I'll show you today was just accepted and published in Human Molecular Genetics. So so all the data is published and we'll be able to look it up. Okay, so the way that my lab, which is at the Mind Institute and UC Davis, approaches the issue of researching duplication 15q syndrome is that we just spend a lot of time with clinicians, clinical psychologists, Shefali, pediatric neurologists, Audrey Thurm, Kate Rowan has been amazing, Dupe 15 Alliance, basically talk to as many clinicians as we can, see patients as much as we can, get as much idea of the syndrome in the patients, and then go back and use the best mouse models that are available. I don't personally make mouse models. We just collaborate with a lot of people to get the best mouse models so that we can do comprehensive behavioral phenotyping for phenotypes that are relevant specifically to duplication 15q syndrome, and then also broader autism spectrum disorders. We look at the animals across development, so basically postnatal day 2 or 4. We're right in there doing various developmental assays. We collaborate with others to look at anatomical and histological phenotypes. This is all with the hope that we're eventually going to be able to intervene by knowing underlying mechanism and therapeutic treatments, and develop therapeutic treatments. And all of this in general goes along with these autism and epilepsy genes, and mechanisms that we have been interested in and are looking at specifically include everything on the side. So we don't really know underlying mechanism, but we've looked at various excitatory inhibitory imbalance, inflammatory pathways, transcriptional regulation, as well as mind connectivity. So it's all on the table as far as Dupe 15 goes. And so we've talked a lot about Ube3a, so I probably don't need to even review this slide at all, nor do I need to review what duplication 15q is for this group. We have focused on Ube3a for the same reason that all the other researchers have. It's the maternally imprinted gene. It has so many important functions, and we know that it's overexpressed in our kiddos. Okay. And so all of its functions, so simple, right? It's extremely complicated. So Ube3a regulates protein synthesis, degradation, regulates diversity of cellular processes, synaptic plasticity, transcriptional coactivator, all of these things. Any one of those mechanisms that I showed you on the first screen could be responsible as a dysfunctional Ube3a. And so the model system, but what nobody has talked about today, and the model system that I'm going to tell you about, is that Ube3a actually has several splice variants in the human and in the mouse. And so we were interested in this idea that perhaps one splice variant in particular would be responsible for certain phenotypes, and maybe other splice variants would be responsible for other phenotypes, or perhaps one would be responsible for them all, or you would need the combination of them to have the full phenotypic syndrome. And so the idea behind this is that if you have one splice variant that is functional and one that is dysfunctional, you could get at targeted treatments. And you could have some Ube3a that's functional and intact while only targeting the dysfunctional one. And so we started to do this collaboration with Larry Reiter and Scott Doe, who made inducible lines of Ube3a overexpression, each isoform specific, and we started out with isoform 2. And by starting out with isoform 2, it's because it's the longest isoform, it has every exon coding, and initially, my hypothesis was around synaptic plasticity, and since isoform 2 localizes more to the cytoplasm, and we could talk about that further later, we thought that that would be most interesting for synaptic plasticity, learning and memory, and phenotypes like seizures. Okay, so I'm going to quickly go over the design of the mouse model, but if you have really detailed questions on it, you would want to ask Larry. So these are the three different isoforms with the, isoform 1, 2, 3. 2 is the longest, has this extra N terminal here. These extra 21 amino acids both have C terminal. And isoform 3, which is probably localized to the nucleus. And then isoform 1 is hypothesized to be an RNA, microRNA. And so here we just have this little cartoon diagram that showed the approach that I just articulated about looking at the isoform 1 overexpressing mouse, which is mostly, it's cytoplasmic and nuclear actually, isoform 2, which is exclusively cytoplasmic, and isoform 3, which is exclusively nuclear. And so the breeding scheme of these mice is really interesting and kind of complicated. And so they are they're a TET-based line. They're TET off, so they don't have to have doxycycline at all to have the overexpression of the gene. And then we breed them with these TTA promoters. And so all the data I'll show you today is from the CAMK2A promoter, because CAMK2A is expressed in forebrain excitatory neurons. And those are the types of phenotypes that we're interested in analyzing, so that's where we started. It doesn't mean that other subtypes aren't involved, and we definitely plan on looking at other promoter systems. So we breed the TTA CAMK2 driver, which is on a B6 background, with this TRE flagged animal, which was on an FEB background. And so all of our phenotypes that we're gonna tell you about today are actually on the FEB B6 F1 hybrid, and I'll show you some control experiments and some extra data on why that's important. So when we say, so when you see all of my graphs and you see wild type versus TRE, TTA, those are the two main comparisons that we made. Although when we did the comprehensive phenotyping, we did actually look at all 4 lines, and I can summarize that data for you on the side because it's a lot of extra data. So the first step when you make a new transgenic model, like Matt Judson just outlined beautifully, is to actually validate it and show that it overexpresses. And so here's the model design again. If we were to give doxycycline, we would stop this binding of the TTA to the TRE, so then we would not have the default overexpression, and so those will be our rescue experiments, which are planned. But initially, we start dox off. And so you can see from these blots here that these were the founder lines where we saw Ube3a2 overexpressed in the cortex. This is cortex specifically with the TTA driver showing RNA increase in the Ube3a isoform 2 and not in the lungs. So this shows that it's central and not leaky. And then that's the Western that shows that it's central nervous system, brain, cortex, not leaky. We wanted to validate that when we did give docs that this system would be reversible, as we were told, or as we had predicted. And so this slide is actually courtesy of Dylan Ritter, who was an Autism Science Foundation fellow and a brother of a child with duplication syndrome. And he worked in Scott's lab for he worked in Scott and Doe's lab for 1 summer and then in my lab for 1 summer, and he did all this doxycycline dosing, and he was amazing, so I just want to give him credit here. But anyway, so what we see here is that in the wild type, the dox and saline look about the same, so we're not having any adverse effects of doxycycline, and that in the saline group here, we have lots of overexpressed Ube3a isoform 2 by this immunofluorescence plot, and then not in the dox-treated mice. And then there's the quantitation of that. And so we validated our system, and we've showed that it's reversible. This is immunofluorescent plot from Jamila LaSalle's labs. So we collaborated with her, gave her tissue so that she could show us that the isoform 2 was in fact localized mainly to the cytoplasm in our CAMK2 drivers. So these first set are from double expression mice. This is the CAMK2 driver alone, the UBE3a stain, the DAPI for the nucleus, and then the merge. So then this is showing that the driver and that these are excitatory neurons, and then here's the flag stain. Stain for UB3A, DAPI for the nucleus, and the merge. So we see that mainly it's isoform 2 is in the cytoplasm. Okay, And then here's where the stuff that I am really good at comes in. So the developmental expression, the behavioral phenotypes that we start off when we take pup calls, you heard a little bit about them from Toro earlier. This is what the pup calls will sound like. They have come in different forms. That one, oh, you know what? I turned off my volume earlier because I didn't want to be rude during the meeting. But the volume's not going up now. Is the AV guy here to help me with the volume? Okay, so the calls, they sound upward, downward, or 2 syllables. Whistle up, whistle down. I don't know how to fix the volume if I need it. Oh, because I'm not plugged in. Thank you. I like the calls. Thanks, Vanessa.
You're welcome.
Okay. So here's what the upwards sound like, and downwards. And then sometimes they have 2 components. Toro showed us earlier that they're not limited to just these 3 types. They have 10 subtypes. But again, as Toro had beautifully articulated, is that we don't really know what the differences in call subtypes mean. We do know that they serve some function as the mom will go pick up the pups as she hears the calls from a speaker. It doesn't actually have to be the mom. The other things that we look at are basically milestones, weight, length, growth in the pups. And so this is what our Isoform 2 overexpression animals look like. So we have less calls over the developmental trajectory from the overexpressing Isoform 2 mice, but normal weight and temperature. And the normal weight and temperature are really important controls, because if they were smaller or if they were cold, they would just automatically be emitting less calls. And so we don't want to be over-interpreting our data. Okay. Gold standard test for anxiety, elevated plus maze. Most people have heard about this like since college. Basically, the mouse doesn't want to be out in the scary open arm areas. They feel more comfortable in the safe enclosed areas, but the animal will come out and explore the open arm and check it out and say, is this scary? Am I at risk? Whatnot. And so what anxiety would look like, or anxiety-like behavior would be spending very low time on the open arm, but the critical control in this is that they have a good number of total entries, meaning they are exploring the maze. They need to get around the task and make sure that they know the difference between the open and closed. And so here we see in cohort, in our first group, lower open arm entries for the overexpressing mice and total entries being normal. So this shows that it's specific to anxiety and not confounded by motor. We reproduce in our lab because reproducibility is a problem. And so here we see the same exact thing in cohort 2. We actually did a cohort 3. So we were very confident this is a strong anxiety phenotype in our mouse model. Another anxiety test was invented by my postdoc mentor, Dr. Crawley and Rich Paler. This is a simple light-dark exploration box. They go back and forth between this open, again, same premise, open area, scary. This is where like a rat would get me, and dark is safe and enclosed and less nerve-wracking. And so the best parameter to measure in this task is actually not the time in the dark, because if you're giving anxiolytic drugs, et cetera, they end up spending more time not mobile. And so it's actually the number of transitions back and forth. We've done this with many different benzodiazepines to check that without sedation or a hyperactivity due to the drug, that the transitions actually are the one that moves. And so again, we see our model had less transitions, suggesting that they weren't exploring back and forth. They have a higher level of anxiety. We see it in one cohort. We see it again. Okay, third test of anxiety is this. This is a really simple test that's used all the time in R&D and pharma, is stress-induced hyperthermia. So this is like your physiological response to stress. I'm not going to show you a picture because literally the task is sticking the probe in the rectum of the mouse and getting its baseline body temperature and then waiting 10 minutes and doing it again because they know they're stressed. They were just probed. And so we see that in the wild types, elevated response in the TRE-TTA, but when you compare the response from the wild type to the mutant, we see a lot higher elevation in the mutant mice, and then again we see this replicated. So 3 tasks, 2 cohorts, a few of them have 3 cohorts, all anxiety related, all have really robust findings in our mutant model. Okay, so I'm not going to talk anything about reversible because we really are just in the middle of these experiments, but I thought that it was really encouraging to see that when we gave the doxycycline to these mice that we just did the blots for, this animation is messed up, but that we did start to see some reversal. So now I just showed you a lot of anxiety phenotype that is robust, reproducible, impressive. We gave doxycycline for 2 weeks at 8 mg per kg, oral injections. This is not the ideal experiment. These are not ends that I would ever publish or ever even really show in public. But I did think that it was kind of cool for the meeting to show everybody that when we gave the doxycycline, which now lowers the Ube3a isoform 2, it's back to what we hope was normal levels. We have the mutant animals still show lower open arm entries on the elevated plus maze, still show lower transitions, still show elevated body temperature, but that the animals with dox came back up. So I just thought that this one slide was encouraging and wanted to share it with you guys. Okay, we talked a little bit about motor confounds. Motor basically confounds everything in mouse behavior, so everybody should be careful and learn that lesson really fast. It's an open field. Animals move around. We check the time that they spend on moving around the course of the whole entire assay. Some people use center time as anxiety. We don't. I could tell you why later if you're interested. Anyway, there was no gross motor differences in the UBE3a mutant animals. So that was encouraging. However, it was a little bit confusing for me because with the strong phenotype of hypotonia, movement, or whatever, I did expect to see some motor. I was happy that it wasn't gross and huge and going to confound all of my behavior assays, but that is what we saw. So now, and this is also preliminary, is what we're trying before we buy this fancy piece of equipment to try this really old traditional test of like walking across a beam to see if there's subtle motor aspects that we haven't gotten to yet. And hopefully this video will flip around for you guys and you can see it like upright and you don't have to turn your head. Maybe it will. I don't know, maybe it won't. Oh yeah, it does. So you can see that his foot slips there. And we have been working through these various widths of beams and rods. Actually, Janine LaSalle's postdoc, Annie Vogel, helped us create this task because they were working on it for their McP2 animals. And now we have it really optimized and it looks really good. And although these are lower ends and we don't see statistical difference yet, we are starting to see trends that there may be a subtle motor phenotype that we should be investigating further. Okay, social. We just heard an amazing presentation by Matt and an amazing presentation earlier by Toro that showed us about sociability in all of our overexpression models. And so this is what it looks like in our lab. We use automated ethovision. We've gone back and manually scored tons of it. This side has the mouse here. This side has the mouse here. You can see that just by the way the tracking system goes. This is the 2015 version of the 3-chambered social approach task. Before I go high horse on 3-chambered social approach, I just want to say that I have been working on this task since 2006, and between me and the postdocs in the Crawley lab and collaborators and colleagues, we've probably seen data on somewhere between 5,000 to 7,000 mice in this task. So I feel really comfortable in what it can do and what it can't do. And we can talk about that later. So these mice are typically social. They spend more time in the chamber with the novel mouse. They spend more time sniffing the novel mouse. They don't have any motor issues moving back and forth between the mouse. So this is not a three-chambered social approach. There is no three-chambered social approach phenotype in these mice. We did it in one cohort. We did it again. We did another task of sociability where we also saw no differences, but I'll get to that in a minute. But what I wanted to say here, and this sidetracks a little bit from Dupe15 model, but it goes into using three-chamber task alone to call sociability or call social behavior in rodents. And I just want to give you this example that we have from our lab that was recently published using the SHANK3 mice. And so most of you are familiar with these other rare genetic disorders that are associated with autism, and one of them is Phelan-McDermid syndrome, which is a SHANK3 deletion. And so the original paper used this three-chamber social approach task and sees, like we've talked about, spending more time, the wild types spend more time with the stranger mouse, and then the mutant animals don't spend more time with the stranger mouse, and thus we call this an autism mouse model because there's no deficits in sociability. And so we worked on this project together where we really wanted to, so this was Dr. Crawley and Mustafa Sahin and myself and Autism Speaks at the time to try to really make sure that deficits that were published in high-profile papers would be able to be reproduced, particularly in the social paradigm. And so we ran our first cohort, and what we saw is that we saw the wild types have our typical sociability scores, and we saw no deficits in females. So what we ran that's quite different from a lot of these high-profile studies is we ran males, and we ran females, and we ran with high power, and we ran in 2 cohorts. And so what you see is if you were to get this first cohort of data where you only ran males, you would see this chamber time deficit and the sniff time deficit. And then you would have an autism mouse model, and you would have a social deficit, and that would be the interpretation. But looking at the females, it was a little more confusing because we saw that they were typically social by this task alone. And then we run a second powered cohort, and we find that everybody was typically social. And so what we had been seeing for a long time, and now this exact set of data showed us, is that this task is really insensitive. And it really doesn't give reliable results time after time. And so I really would encourage anybody that's interested in animal models or studying animal models of autism to be careful on making broad interpretations with just using three-chamber alone, which nobody in this room has done. That's my 2 cents.
How are these mice housed? Are they single housed or group housed?
Absolutely not single housed. That would greatly affect sociability.
And what's the genetic background?
Of Fang SHANK3 mice, B6J. But I'll talk to you about FVBs later. I got something else in here for you. Okay, so this was that task that we just talked about. So I just want to say, like, if we could just be skeptical or, we're scientists, like we want to have good practices, standard practices, and a cautious bit of optimism for social behavior. Okay, and one other thing about this, and then I'll get off of it and we can talk about this later, is that it's three-chambered alone is not sensitive. We've published many reviews on how we think it should optimally be used and to just use 2 or more assays, which I said everybody has been doing. This I'll skip because I want to get back to my overexpression mice. So again, normal sociability in this direct dyad interaction task, which you don't need to see the video for it. And then these videos are a bunch of autism-relevant behavioral assays, grooming, jumping. This mouse jumps in the video when it works. These mice on the bottom circle. Maybe when the video works, they circle. Can you get it? Yeah, so in this video, it's hilarious. I won't even have to tell you who the mutant is. They just circle. So mice do all these unique repetitive behaviors that make them good for studying autism models, but our overexpression mice had no repetitive behaviors, none of these listed, no social deficits. Okay, learning and memory. So fear conditioning, very simple. It's like Pavlov. They go into the area. It smells like vanilla, the grid floor. They get shocked. They remember that it smells like vanilla and that it's the lights on and that they get shocked. And then you bring them back and it smells like vanilla and there's the grid floor and there's the light on. But they don't get shocked, but yet they freeze because they remember being there. In cued, they go back to this chamber. It's triangle. It's dark. It smells like orange. They don't remember that that's the context that they got shocked, but then you play the tone that you had played with the shocking and they remember it. So our model mice had normal level, so this is the first stage where they get trained, tone shock pairs. Our mice had normal levels of post-training, which is good, meaning that they could freeze and they weren't unable to freeze. But then they had deficits in the contextual phase of fear conditioning, which would suggest a learning deficit, but not in cued, although this is borderline, but they didn't have deficits in cued conditioning, which is not unexpected as the neuronal circuitry that underlines cued and contextual is different. And just to keep in my theme of reproducibility, this exactly replicated where we had context deficits and not cued. And then this is my last thing, and this is the coolest thing. So those 4 genotypes that I told you about at the beginning, everybody's number one question is, do the mice have seizures? Do the mice have spontaneous seizures? Everybody wants to know that. We don't think we saw that the mice have spontaneous seizures. However, because we had 4 genotypes in every single litter, and some animals did die, we're not positive that we don't have spontaneous seizures. We're going to have to analyze that now with our new system more comprehensively with telemeters over many, many hours. But what we did do for now is do penicillin tetrazyl injections and look at behavioral and EEG seizures afterwards. And so this was our first cohort where we looked at the latency to the first generalized clonic seizure, which would be loss of writing reflex. The model did it much faster. Full tonic extension or death, they did it much faster. This is a representative EEG trace from when we were using tethered EEG that showed us that the wild types had seizure here, continued to go on and live. Our model mice went into seizure much faster, much more robustly, and then died very quickly. Again, all of that replicated in a completely separate cohort with PTZ. This is my last slide. I just wanted to show you guys this video. Nicole, my graduate student who's up here, worked really hard on creating this video for us. This is our brand new system that we wanted to show off. We're using these untethered, so let me play the video before I get too far in anything. So this is our untethered system. You could see right here, this guy has a little implant here. It's a telemeter, just like a barcode would be at the supermarket. And so he or she can live in a home cage. He can live with other animals, so he doesn't have to be isolated. We gave him PTZ. He had the Straub's tail, like the beginning of the seizure, and then he goes on to have more severe seizures. And then simultaneously, you can see it not only through the behavior, but we have the corresponding EEG and EMG that we're collecting. And we're doing these in our model mice right now at the Mind while we're here. And so since I'm running on time, I will not get into this background strain stuff, but I would encourage everybody to see Abe Palmer's lab's paper that just came out in Neuron about moving on from using single strains, and everybody's talked about this for a while, but just looking at something on one strain alone and not more than one, which was, at first I thought I was gonna die when I was looking at hybrid mice for this cross, when I was like, oh my God, they're FEBB6, what are we gonna do? And we started back crossing, and I wanted to really get everything on chondrogenic B6. And then this Neuron paper came out, and I'm like, wait a minute, maybe I didn't wanna get everything on B6. Because this paper actually shows that background strain is more of a confound with genetics or with one gene than sex. And so we really need to stop looking at the same old B6J or all one inbred strain line and think more broadly about that. And there's 2 things that could have confounded our data here, which was the hybrid, like I just said, Or there was one paper in J of Neuroscience that suggested TTA toxicity, but we ran 2 comprehensive cohorts on all of the behavior that I showed you here today to show that none of those were confounding effects. And if you want to hear more about that, I'm willing to talk about it later. Reproduce, here's our paper. These are my conclusions, that we generated and validated the mouse, that we have really nice phenotypes that highly are analogous to and clinically relevant. So we do think Ube3a is important. We think Isoform 2 is important. And we hope to be able to show you the rescue stuff soon and then have all of our data on Isoform 3. And these are my collaborators, Scott and Janine. And this is my whole lab, especially Nicole. And Jacob, I'll show you the imaging of all this mouse stuff. That's it.